Okay. Start. Start.
Hello everyone, welcome to Extra Marks NEET YouTube channel. As you all know, the exam is fast approaching on May 4th, so we are here to help you to make the last minute revision. In this session, the Crack the Code Botany, we are going to take certain images as well as flowcharts from your 12th NEET Botany, and we are going to understand the topic. Then we'll also take some questions which has appeared in previous year exams and we'll be discussing on about them. Now let us start the session, and I want each and every one of you to be active in the comment section on the questions which are going to pop up on the screen. Now the first topic on today's session is stamen, the transverse section of young anther. As you all know, the male reproductive structure of the flowering plants is called androecium, and androecium can have multiple stamens. Sometimes it could be 1, sometimes it could be 10 based on different plants. Now the pollen grains are going to be synthesized inside this particular androecium itself, stamens. Now in this stamen, we have two parts, one is called as a filament and the other part is anther. When you take a transverse section of this anther, now when we take a transverse section of this anther and observe it under a microscope, this is how a typical anther section looks like, a bilobed dithecous anther which is found in majority of the flowering plants. Now the anther wall consists of 4 layers. The outer layer, which is called as epidermis, as the name states itself, derma means skin, which starts from outside, and it is a single layer protective. The second layer, it's called endothecium, which basically helps in dehiscence of the anther. So later on the pollen grains are going to be released outside. Followed by the middle layer, which is made up of 3 layers, which helps in storage because it is basically made up of parenchymatous tissues. And finally, the most important layer, which we call it as a tapetal layer, which provides the nourishment for the growing microspores later on, which are going to change into pollen grains and released outside. A similar type of questions are asked in NEET exam. For example, in 2013 in NEET exam, one question was asked, which one of the following statements is correct? Option A, endothecium produces the microspore. Option B, tapetum nourishes the developing pollen. Option C, hard outer layer of pollen is called intine. Option D, sporogenous tissue is haploid. Please write the answer in the comment section. Yes, absolutely right. The answer is option B, the tapetum cell, which helps in the nourishment of the developing pollen grain. Many times they can also ask question on endothecium also. What is the function of endothecium? The dehiscence part. And sometimes they can also ask question about what is the function of epidermis, as the name states itself, derma, which gives the protection itself. Okay. Let us take the next topic. The next topic is the male gametophyte, which is pollen grain itself. Now once the pollen grains are developed, they mature and they form the structure. Now pollen grain is typically made up of 2 layers. The outer layer is called as a exine, which is one of the hard layer which consists of a chemical called sporopolanin, which is one of the highly resistant chemical substance found in the plant kingdom. Followed by the inner layer is called intine, which is made up of most common chemicals which are found in the plants, that is cellulose and pectin. There are also certain pores found throughout the pollen grains. Sometimes it could be few, sometimes it could be many. We call them as the germ pores because that is the place where the intine protrudes during the germination. And majority of the time in flowering plants, close to more than 60% of the pollen grains are found in 2 cell stage. One we call it as a large vegetative cell, which has a asymmetrical nucleus, which is like uneven in shape. Then there is a generative nucleus, which we also call it as a male gamete nucleus. In 60% of the flowering plants, the 2 cell stage is very common. In remaining 40%, it varies up to the 3 cell stage. Now let us see the question which has asked on the male gametophyte in the previous year. In 2024 NEET exam itself, there was a question, pollen grains remain preserved as fossils due to the presence of epidermal layer, tapetum, exine layer and intine layer. Please write your answer in the chat section. Yes, absolutely right. The answer is exine layer, all thanks to that highly resistant chemical substance sporopolanin, which keeps the pollen grain alive for many years. There are some plants where the pollen grains are excavated even after 10,000 years. If you talk about one of the plant called as a Lupinus articus, where the pollen grain were excavated after 10,000 years and plants were successfully generated. The recent discovery also goes back to 2000-year-old Phoenix dactylifera, where the seeds are excavated from those, not the pollen grains, the seeds are excavated from those particular plants. Next, we would like to talk about ovule, or we also call it as a megasporangia structure. As you all know, the ovule is the structure which is going to change into seeds, right? Now, typical ovule has a chalazal end and a micropylar end, and it starts from xylem, which goes and connects to the ovary part, right? Then there is a funicle closer to the micropyle. Then there is a completely filled sporogenous or nutritive tissues in the inside, which is called as a nucellus. And in majority of the plants, it is bitegmic, outer integument as well as the inner integument. There are some plants where ovule is also made up of a single integument. There are some plants where integuments are also absent. We call it as a ategmic also. Now let us take a question from this, which was asked in 2024 NEET. Which part of the ovule stores reserve food material? Nucellus, integument, placenta and funicle. Yes, absolutely right. The answer is option A, nucellus. From nucellus, the nutrition is given to one of the nucellus, which takes all the nucellus nutrition and it grows, and we call it as a MMC, megaspore mother cell. Later on, it undergoes what we call it as a megasporogenesis, which results in the formation of a megaspore. And this megaspore later on changes into megagametogenesis, which results in the formation of a embryo sac. Okay, let us take the next question. The next question is about embryo sac, a female gametophyte. As we all know, the single monosporic embryo sac develops in majority of the flowering plants. A single monosporic embryo sac consists of 8 nucleated 7-cell stage. Let us see what are all those structure. So the first structure I would like to talk from the chalazal end is 3 antipodal cells, and all the cells which are found here with the nucleus, they are all haploid in nature because it is a product of a meiosis. And there are 2 cells which are found towards the micropylar end, we call it as the synergids, which also has one more cell closer to which we call it as egg cell, and both the eggs and synergids together they are called as egg apparatus. There are also specialized structures which are found, a cellular structure which is found inside the synergids, we call it as a filiform apparatus, which basically helps in guiding the entry of the pollen tube inside the synergid during the fertilization process. And there are also 2 central nuclei, or we also call it as a polar nuclei, without any kind of cell covering them, but there is one overall cell which covers all these cells together, which we call it as a central cell. Now, there was a question in 2021 which was asked on this particular diagram, and the question was, a typical angiosperm embryo sac at maturity is. A typical embryo sac at maturity is A, 8 nucleate and 7-cell stage, B, 7 nucleate and 8-cell stage, and C, 7 nucleate and 7-cell stage, and D, 8 nucleate and 8-cell stage. Please write the answer in the comment section. Yes, absolutely right. The answer is option A. So the typical angiosperm embryo sac, which we call it as a monosporic polygonum embryo sac, which consists of 8 nucleated 7-cell. That means there are certain plants which also produces more nucleus and more cell, but this is the most common type of embryo sac which is found in the flowering plants. Okay. Now let us take the next topic. The next topic is a false fruit, right? Now what do you mean by a false fruit? To understand this, first we need to understand where exactly the fruit comes, then we will understand what do you mean by false fruit. As you all know, in flowering plants, the ovary changes into fruit and the ovule changes into seed, right? Now there are some plants where the fruit is not developed from the ovary. Instead of this, there are other parts in the flower which are involved in the development of the fruit. Now the simple example we can talk is apple, strawberry, right? So basically what we are eating in this particular fruit is not a ovary, instead we are eating the other part of the plant. For example, you can see the part where we are eating the pulp which we are eating, it comes from a structure called thalamus, or we also call this as a receptacle. Now this type of fruits are also called as false fruit. Now let us see a question which was asked on the above topic, which was on 2022. Which part of the fruit labeled in the given figure makes it false fruit? So there are parts A, B, C, D, but in this only one part directly tells this is a false fruit. A, C, thalamus, option B, D, seed, option C, A, mesocarp, and option D, B, endocarp. You can all write answer in a comment section, please. Okay, one of you have asked, sir, what is orthotropous ovule and anatropous ovule also? Just a minute, after this I will be telling about that. Yes, absolutely right. The answer is option A, thalamus, where the false fruit develops. So based on the position of the ovule, or the structure of the ovule, in that we have structure called chalazal end, micropylar end, as well as the funicle end. So based on this structure, how they are positioned, ovule is divided into different types like orthotropous, anatropous, different stage. So orthotropous is also one type of ovule where all the positions lies in the same line, whether it comes to chalazal end, whether it comes to micropyle and funicle, which is more common in gymnosperm. Similar way in angiosperm, the most common type is called as an anatropous kind of ovule. Okay, next. The next topic I would like to talk is symbols which are used in pedigree analysis. So during analysis of various disorders, genetic disorders, we use different types of symbol to see whether a particular genetic disorder has been passed on from one generation to another generation. Now this is what we call it as a pedigree symbols. For example, to represent a male person in a particular family, we use something like a square rectangular kind of shape. For a female, we use a circle. And suppose if we are not able to identify the sex of an organism, for suppose if baby sex is not determined, we call it as a sex unspecified and we use this symbol. And if any one of this individual is affected from particular disorder, we have to shade them. Like when you are drawing, we will be using like this empty box, and suppose if this person is affected, we will completely shade it. We'll completely shade it just to show that this person is affected, right? And if a male and female, they're married and the mating is there, they're reproducing, we use this symbol, just they are mating. And if the mating is happening between the closely related relatives, like consanguineous mating, we call, like they are related either from the father side or from the mother side, somewhat there will be relatives, and we will be using the double symbol, and this is called as a consanguineous mating. And to show the children below the parents, we can use the symbol like this reverse T order, where you can write how many children are there. Suppose for this parent there is a one son and one daughter. If there are multiple, we can also extend this and we can also draw like this. And suppose one of this child is affected, here you can see it has been completely shaded to show this child is affected or disease is carrying. And sometimes we don't know the sex of a child, we use this symbol, right? Sometimes what if it is twins, triplets, we have to show. So inside that we can write a number, how many childs are there. Okay. Now sometimes in your exam they will use this kind of symbols and they will ask you to tell what exactly the meaning of that symbol. Now let us take one question which came in PYQ 2023. So the question was, which one of the following symbol represents mating between relatives in human pedigree analysis? Option A, option B, option C, and option D. Please write your answer in comment section. Yes. Answer is option C, where you can see there are two lines which are connected between a male and female. So that is your right option. You can say this is a consanguineous mating or closely related kind of mating. And here you can see all of them are affected. And here you can see a mating between a male and female, but they are not close because there is only one symbol. And here you can see a father and a mother having a child who is a son because of the box and he is affected or deceased. Now the next topic I would like to talk is about a chromosomal disorder. Now you can see there are two diagrams which has been given here. One is a man diagram and another is a female diagram. So one man is suffering from a Klinefelter disease, that is basically he has one X chromosome. So a normal man has a 46 chromosome, right? Here he has one additional X chromosome attached to the last allosome. So instead of he having XY, he has XXY. And the other image is about a woman who has Turner syndrome, where she is supposed to have XX chromosome, but here she has one less X chromosome, so she has only one X chromosome, right? Now here if you see the characteristics between them, there are some characteristics which is little different than the normal human beings. For example, if you talk about the Klinefelter syndrome, here you can see this particular person as a little bit enlarged breast character, or we also call this as a development of gynecomastia, where there is an enlarged breast characteristics are there, and there is something called as a testicular atrophy. This person will not produce any kind of sperms, and there is a poor beard growth is observed. So these are some of the character, and he also has the feminine low pitch voice. Same way in the Turner syndrome, where there is one less X chromosome, this female is missing some of the feminine characteristics like rudimentary ovaries, which doesn't produce any kind of eggs, underdeveloped breast, and widely spaced characteristics of them, webbed neck. All these characteristics are observed. Similar way, there is also Down syndrome, where that particular person has a trisomy condition, where on the 21st chromosome they have a additional chromosome, right? And they also have a characteristics like creased palms, right? And their tongue is protruded, furrow tongue protruded, similar kind of characteristics. Now in 2019, there was a question which was asked on gynecomastia, which is related to the one of the disorder. The question is, what is the genetic disorder in which an individual has an overall masculine development called gynecomastia and is sterile? Option A, Edwards syndrome. Option B, Down syndrome. Option C, Turner syndrome. Option D, Klinefelter syndrome. You can write answer in the chat section. Yes. So answer is option D, Klinefelter syndrome, which is basically typically utilized by this one particular terminology called gynecomastia, that is enlarged breast in the men due to the presence of one additional X chromosome. Let us go for the next topic. The next topic is from the structure of DNA, which is the Chargaff's base pairing. As you all know, Erwin Chargaff gave us a base pairing rule. You know that A always joins with T with the double bond. At the same time, G always attaches to the C with the triple bond. And as you all know, if there are 10A, there is going to be 10T. If there are going to be 10G, there is going to be 10C. So they will be always in the equal. So sometimes questions on this particular base pairing is also asked in your NEET exam. For example, in 2021, there was a question which goes like this. If adenine makes 30% of the DNA molecule, what will be the percentage of thymine, guanine and cytosine? A, T is 20, G is 25, C is 25. B, T is 20, G is 30 and C is 20, and C, T is 20, G is 20 and C is 30, and D, T is 30, G 20 and C 20. Please write your answer in the comment section. Yes, answer is option D. As you all know, the total, what is the total percentage of adenine? The thymine is always going to have the similar, right? So you don't even have to worry about G and C in this particular question. As you know, if adenine is 30% and thymine has to be 30%, so directly you can tick on T without knowing the guanine and cytosine. So answer is option D. Sometimes they can ask a particular organism name in this and they will ask you to calculate the percentage. Always remember, doesn't matter which organism name they are going to use. When the calculation comes, you just have to apply the Erwin-Chargaff's rule, which always remains same, which always going to remain the same. So adenine, the thymine will be same. Guanine, the cytosine is going to be the same. Next. The next topic I would like to talk is packaging of DNA helix. See, in human beings, the DNA length is approximately 2.2 meters, right? So imagine in such a microscopic cell, this DNA needs to be packaged and compactly arranged in one particular place. So what exactly this DNA is going to use to get it packaged itself? As you all know, DNA is negatively charged. As you all know, DNA is negatively charged. It is going to take the help of certain specialized proteins, which we call it as a histone proteins, which are basically positively charged, and this particular histone proteins comes in contact with the DNA. Suppose if this is the protein, now DNA goes around this proteins and forms this beads like structure. It goes around the protein and it forms like this beads like structure, which creates a compact structure which we call it as a nucleosome. So there are 9 histone proteins, right? They all come together to form this structure which we call it as a nucleosome. Now some questions are always based on this histone proteins also, how it is arranged, why it is arranged, and etc. Such question was also asked in 2017 NEET exam. The question was, the association of histone H1 with a nucleosome indicates basically what? A, DNA replication is occurring. B, transcription is occurring. C, the DNA is condensed into chromatin fiber, and D, the DNA double helix is exposed. So what exactly do you mean the last H1 protein which goes and blocks everything once the nucleosome is done? What exactly it indicates? You can write answer in the chat section. Yes, absolutely right. Answer is C. So basically when DNA gets coiled around this histone protein, they get condensed. The chromatin fiber get condensed and H1 protein is acting like a block. So this DNA will not roll back, right? So it is basically showing the chromatin fiber is highly condensed. So based on this condensation, we can again divide this chromatin fiber into euchromatin and heterochromatin based on how much stain also it takes. But basically it's on how they are condensed, easy condensed or highly condensed. So that is called heterochromatin as well as the euchromatin. Okay. Now let us take the next concept. The next concept is genetic code. As you all know, the codons are responsible for coding different types of amino acids, right? So there are total 64 codons. Out of this, 61 codons code for 20 different amino acids, while there are 3 codons which we call it as the stop codons, which doesn't code for anything, but they help in during the protein synthesis or during the formation of transcription during the mRNA formation. Otherwise, the remaining 61 codons are responsible for coding for different types of amino acid. A very easy way to remember is already been given in your textbook. You can see, you can write UCAG, or you can change this order also. There is no rule you have to write UCAG only all the time. You can say UACG also can be written, but make sure whatever you are taking the codes for every box here, the same thing remains on the top side. But on this side, you can see on every box UCAG has been written. So one on the right side, you can see UCAG for one letter for every box. On the top also one letter for every box. And on the left side you can see UCAG for every box. And now take the first letters of everything like U, and U. And again U and C. So like that if you write, you can remember all the 64 codons. In this, other than these 3 stop codons, there are 2 other codons you need to remember in the exam point of view. One is UGG, which codes for tryptophan, because only one codon codes for one. And next is methionine, which is AUG, which has a dual function. One, it produces methionine as an amino acid. Second, it is also responsible and act as a start codon. That's why we call it as a dual function. Now let us see the question which was asked in 2017 PYQ NEET exam. So suppose if there are 999 bases in the RNA which codes for 333, because as you all know, three letters which gives us one codon codes for one amino acid. So 999 divided by 3 will give us a 333 amino acid. And the base position at 901, so exactly the 901st base position is deleted such that the length of the RNA becomes 998 bases. Now how many codons will be altered? A 1, option B 11, option C 33, option D 333. Yes. Option answer is option C 33. As you all know, up to 900 there is not going to be any kind of changes, the codons will be same. Since the only 901st codon is affected, so after that whatever the amino acids are going to be codon, only those are going to be affected. So answer is option C. So 33 codons which is going to result in the formation of different amino acids are going to be affected, not before that. Next. The next topic we would like to talk about is the central dogma. This is a simple image which has been a simple flowchart kind of structure which has been given in your textbook. Often students tend to ignore this, but there has been numerous exams where this question has been asked. You can see in the first structure, they are given the DNA and they put one arrow mark which is circling around the DNA, which basically says the replication is happened. DNA is capable of replication, right? Now the DNA transcripts and forms mRNA. We call it as a transcription, and RNA goes under a translation which helps in synthesis of a protein. And in some organisms where RNA is the genetic material, their reverse transcription will happen where first RNA changes into DNA. Later on the transcription and translation will take place. Now this same diagram could be asked in your exam, and some questions can be framed in such a way. For example, in 2017, the complete central dogma was given and these are the options they told. And they asked what exactly A, B, C, D represents. What exactly A, B, C and D represents. Please write your answer in the chat section, please. Someone has asked, sir, can you teach a very difficult topics? We can, going ahead. So this is a kind of a fast track of certain diagrams from your textbook. Going ahead, please suggest some topics, we'll be teaching them also. Yes, absolutely right. Answer is D. As you can see, A is a replication, B is a transcription where DNA helps in synthesizing the mRNA, and finally a translation, C, and finally the D is protein synthesis. Okay. Let us take one more question. The next question is from a concept called lac operon. As you all know, there are different types of genes which is found in our body, right? And sometimes some genes comes together to do a particular function, and a group of genes which are doing a particular function, we can also call it as an operon system. And there are two types of genes, functional genes and structural genes. So one which basically tells what to do while the other genes will do it, right? If you talk about certain organisms, in those organisms body, there is a system where it switches on and switches off whenever it is required to save certain energies. In such operon system, there is an operon called lactose operon, where there are three structural genes which basically are Z, Y, A, which goes and undergoes transcriptions, and it goes on synthesizing three enzymes called beta galactosidase, permease, and transacetylase. So different operons will have different structural genes. They give different types of enzymes for their own functions. Now sometimes they will ask what exactly this genes will give us, what exactly I or R or P represents in your exam. In 2022, such question was asked in the match the list, match the following. And you can see in the list 1 they have given the definition like what exactly this word translates, and in the list 2 they have given certain enzymes. We need to match the following them, and option A, option B, and option C and option D are here. So just by knowing 2 of them you will be able to answer here. Please write answer in the comment section. Yes. So the answer is option C, where you can see different enzymes. Like when you talk about Z, it codes for beta galactidase, right? So ZYA, that is the order. So I hope you all enjoy the session. If you would like to have more such videos and more difficult concepts in upcoming days, please like and subscribe Extra Marks NEET YouTube channel. Have a nice day. Take care.